next and last step in this part, and then we're done for today, at least with the lecturing. We need to talk a bit about this FDR thing. What does it really mean? What's a false discovery rate? How do you determine it? And afterwards we have hands-on where you can look into some aspects of that. And I can give you only brief glimpses of that. This is a complex topic, but I hope I can get you started with it, and then we can have some discussions and see about the details. When we do a database search, then this database should contain all the protein sequences that are potentially in the sample, but it shouldn't contain anything else. We will see that there might be an exception to that. From the database and from the enzyme that you selected, you can then generate all hypothetical candidates. Apart from the expected proteins, you should also include some other things that could still be in the sample, even though they might not be in your biological sample initially. And these are contaminants, keratins, for example, or trypsin, of course, because trypsin is still in your sample, and you might want to figure out that some of these spectra come from trypsin. What happens if you don't do that? Yeah, then you can get hits to other proteins, and it could seem like these proteins are in there, even though what you have is just trypsin, which is kind of boring. But, you know, it's there, so it should be included. So try to make sure that whatever you have in your sample is covered by the database, and we discussed that earlier already. Try not to be super inclusive, yeah, because if you put the whole world in there, then the odds of just having random mismatches will increase as well. Databases can also be designed in a way that they give you a good idea of this false discovery rate, so the rate by which you have incorrect identifications, and that is the idea of target-decoy search. Target-decoy databases are based on a very simple idea. You put in sequences, like reverse sequences, of which you're sure that they can't be in your sample. Why the reverse? Well, the odds that the same sequence occurs just in the inverse direction is pretty slim. And this way you would still include sequences also with the same composition, with the same amino acids, with the same length, something that looks very much like your original sequences, but of which you know it can't be in there. And then you can search the spectra twice, or you can do one search and do that again against the combined database. And we can use that to identify the trust that we have in individual identification. So how does that work? Assume you search a spectrum, an experimental spectrum, against a database where you included your organism, say yeast, and then you take all the proteins and read the sequences from the back. None of these proteins should be found in your sample. So what will happen is that your search engine will assign scores to the individual peptide candidates, and hopefully the true identification comes from one of these target proteins. So we now decide target, and the reverse wants the decoy proteins. They look like proteins, they're decoys, very good decoys, but they should not be found. The other hits will hopefully get scores that are not too shabby either. We sort them all by scores, and then you will see that eventually you start getting decoy hits. Hopefully at scores that are much lower than your top hit. Now, what does that tell us? If you go down to a score that corresponds to this hit, then amongst the hits that you have up to this point, you would have one potential decoy hit. That seems to be the score where you start seeing decoy hits. You would like to avoid that, but you will never be able to get rid of these incorrect hits altogether. We've discussed it earlier. There can be random hits, there can be things that are missing in database. You will always have incorrect identifications. But what you can do with this approach is you can try to estimate how many of your hits occurred by random and how many hits should be correct. How is that done? Well, you can think of these scores that each of these peptide spectrum matches has as coming from two distinct distributions. If you look at all the PSMs, all the identifications that you have, then each of them has a score, and the frequency here tells you how often do I see this particular score. For all the incorrect PSMs, you get a score distribution as well. They don't have the same score. Otherwise, the incorrect ones would be easy to identify. You would just expect this score on average to be lower than the scores of the correct identifications. So basically, you have like two distributions that are sitting on top of each other. This part here that contains your correct identification. You get only hits from the true identifications. And you could argue, well, I choose a threshold where I have none of these identifications, where nothing can go wrong. That's like this score of 40, or certain arbitrary score that you make up just by looking at the spectra. But you would lose all these guys here, which are still correct identifications. And it might actually depend on the question that you have, which of these FDRs is acceptable for you. Don't be confused. Don't let anybody tell you that 1% FDR is what you have to do. Don't let anybody tell you that 5% FDR is what you need to do. It depends on what you're looking for. If you want to search as many identifications because you're trying to form a hypothesis, then it might be totally fine to have 10% falsely identified peptides, because you probably are going to do some downstream validation anyhow, and then 10% incorrect identifications is much better than missing an interesting candidate that has biological meaning. So there is no gold standard. You use an FDR of 1% or 5%, but depending on where you set it, if you set it here, you will have a larger number of incorrect identifications than if you go to a higher score. Now, when you use this reversal, then fundamentally what you would expect is that the number of decoy hits at rank 1, so basically at the top of the list, should be small. It's never going to be 0. I just plucked these figures here out of the paper that describe that. If you do a standard search, you would expect that for many cases, the top ranking hit comes from the target because it's the right hit. There shouldn't be any decoys. But of course, you will have decoys, just not as many as targets. Now, you can do a simple trick. You shift the precursor. Precursor shifted search. What does that mean? You just assume an incorrect path. Now, your database search engine would go looking for candidate, but all of them should be wrong. Yeah, because it's off by a couple Dalton. It should not give any good candidate. If you do that, you see that on the top rank, you see as many targets as you see decoys. So it's randomly hitting the two databases, and that's why it's essential that the target and decoy database have similar size, or identical size, I should say. And that's easily done by reversing it. Yeah, the number of proteins won't change. That won't change. But looking at this graph, you can see where your decoys are and where your targets are, and you can adjust this false discovery rate to find as many as you would like. How do you compute the false discovery rate? Well, for a given threshold that you choose, you just check out how many false positives are. False positives are what? Decoy hits. Yeah, they are false positive identification over the number of true positives plus false positives. That's the number of total identifications that you have. Now in the literature there are different ways on how to compute that. They differ only slightly, but not really in the results. The OpenMS tool false discovery rate uses this one, the one suggested by Lucas Schell. And using this simple trick, you can force arbitrary search engines to compute false discovery rates. Just by doing this ad hoc and by trying to recompute the false discovery rate after the fact. Note that this is the false discovery rate on the protein spectrum match level. So if you have 1% FDR, what does that mean for the number of correctly identified proteins? Well, that's one of the big issues, and how to this question, we're trying to answer that. The answer is a lot more, but it depends on the context what it really means. It has to do with a protein inference problem, because so far we ignored how we actually get to the proteins. If we do proteomics, we fundamentally don't care about peptides, but so far all I have been talking about were peptides. I haven't really said how do you get from the protein to the peptide, but to many of you that might seem, from the peptide to the protein, for many of you that might seem trivial. If you know where the peptide comes from, then you know the protein. Unfortunately, it's not that simple. So what's going on there? Identification methods pull out these PSM, these peptide spectrum matches out of the database, and you can compute the FDR on that. And an FDR of 1% would mean that 1% of the PSMs are incorrect. But how about the proteins? Now let's look at identifying proteins based on a peptide sequence, because that is not so trivial as well. So each PSM above a threshold corresponds to a match of a spectrum to a peptide and a match of a peptide to a protein. The protein somewhere. We put it out of the database. We would not identify anything that is not in the database. So you could argue, well, I know the protein. If I've seen the peptide, I found it in a database search, then I should know the protein. What can go wrong there? Yeah. So we need to talk a bit about uniqueness. And it has to do with length distribution. So what is shown here is the number of peptides that were detected in a proteomics experiment. And that's this solid curve here. And you can see there is a maximum at about 10 amino acids. And the other curve is what you should expect if you do an in silico digest of your protein database. So why the difference? What's going on there? So apparently all the left-hand side here, that is all missing. And why is that? Could you speak up a bit?
Shorter peptides are just going to, they're not going to be retained by the column.
Yes, that's one of the problems. Yeah, we usually filter out these masses. Why don't we care? Why don't mass spectrometers care about peptides of length 3? The odds of finding this sequence in a proteome are pretty good. You will find it everywhere. So if you know that the sequence is AKK, what do you know? What did you learn about your protein? Nothing. So we filter at every step of the sample preparation, and also in the mass spec, we filter these guys out. They tend not to be unique. But we can look a bit closer at that. if we are doing proteomics, we really want to identify and quantify proteins, not peptides. So we need to figure out what is going on with unique peptides and with non-unique peptides. And by unique, I mean occurring more than once in the database that we were looking for. So that means uniqueness is not something that is sort of globally defined for a peptide, but it's always in the context of a database. And the larger your database is, the less unique your peptides are, because the odds of just containing that by random are increasing. So there are different reasons for this non-uniqueness, chance hits. If you have a length of a peptide of length 3, then at some point you will have to have it in the database just for statistical reasons. But there can also be different isoforms. Why different isoforms? Well, if you have two isoforms of the same protein, then the majority of the exons will be shared between these two isoforms. And that means we have the same entry twice in our database. But we also have conserved regions in protein families where there is an evolutionary reason to maintain a particular sequence. So let's look at databases. So what's shown here on the left-hand side is the total number, the number of peptide sequences for different protein databases. And in black, we have the total number of tryptic peptides contained in the database. And in gray, the total number of unique tryptic peptides. And as you go from reference data, well-annotated reference databases like SwissProt, RefSeq, to the more redundant databases like Entrez that contain multiple sequences, you see that the number of unique peptides stops growing. Instead, you get all these other redundant peptides that are still there, but are repeated in the database over and over again, which is why it makes not a lot of sense to go to these redundant databases, because all it does is really increase your search time. There are different types of uniqueness, and this graph tries to illustrate that. So you know, gene structure can be pretty complex. So here we have an example where we have 4 genes, and highlighted in green are the exons and gray the introns. And you know, different splice variants arise by combining different exons in different manners. So there are going to be peptides that occur only once in the whole genome, like this red guy here. It doesn't occur anywhere else. And that's sort of the ideal case. If you found this particular sequence, then not only do you know that there is a gene product from gene 1 in your sample, but you also know that it has to be this isoform, because all the other isoforms of protein 1 do not contain it. So once you found that, you know we have protein 1.1 in our sample. We can't make a statement at this point about protein 1.2. Just this peptide tells us that story. You can have ambiguousness at plenty of other levels. So what happens with this brown one here? So that is ambiguous when it comes to the protein level, but ambiguous when it comes to the isoform level. So if you find this guy, you know you have protein 1 encoded by gene 1 and by no other genes, but you can't keep the two isoforms apart. So that's you have to figure that out differently. If you see this guy and this guy, what do you know? Well, you know that you have gene 1 in there and you have protein 1.1, but seeing this guy could mean that you have only this guy or that you have both of them. That would not be distinguishable. So there are different levels of uniqueness up to sort of this protein here that occurs in multiple genes. So if you see this guy, you know that you have gene 1 or gene 2 or products from gene 1 and 2 in there, either one of them or both, and you can't keep the two apart. So there are different types of ambiguousness there. And that brings us back to the question of how many of these isoforms are there. So when we look into NextProt I looked at release 3, and there were 20,000 human proteins and 39,000 sequences resulting from alternative isoforms. So we have, on average, 3 different splice variants for each of these protein sequences. Many of them don't have any isoforms, and then there are some that have like 16, 17 different isoforms that often differ only in small parts of the sequence. Resolving these isoforms is possible if you have peptide evidence that distinguishes some of them. Otherwise, you're in trouble. And this here is one example, PDE9, and that is, these are the 16 documented isoforms of PDE9. And you can see that if you're only interested in the presence of PDE9, then any of the peptides here in this C-terminal part is going to be sufficient. But you can imagine that keeping these isoforms apart is going to be nightmarish if you know that these isoforms matter. And often enough, for some proteins, isoforms have very different biological function. And in this case, you would need to make sure that you really have enough coverage or possibly use other methods to complement your proteomics data. The coverage is often not so great. This here is an example. Highlighted, there are two isoforms here, and highlighted in black are actually the peptides that are seen. And you can see the coverage is not so great in proteomics. Oftentimes you see just 1 or 2 peptides per protein, and then it becomes really hard to keep isoforms apart. The other thing is we often have large protein families, and in these families we have evolutionarily shared peptides. And again, you run into the same problem. Can we keep them? So in eukaryotes, the number of unique can often drop below 50%, and that makes it very hard to really figure out which proteins are. There's a very simple idea to deal with that, and that's Occam's razor. You might have heard of that. It's a concept from philosophy. Basically find the smallest set of proteins explaining all observed peptides. This is not, I'm not saying that this is the right way to do it, yeah, but it's the most obvious way to do it. If all peptides mapping to one protein family can be explained by a single protein, then it's quite likely that only this protein is present. Occam's razor is the simplest explanation for what I'm seeing. And you can argue with that, but it's a simple way to go about it. Let's look at a few scenarios there. So if we have 2 proteins A and B, then we can observe possibly these 2 peptides and these 2 peptides, in which case they would be distinct. Proteins do not share the peptides, in which case if you see these guys, you can clearly explain away what's going on. You know, based on the evidence, that these 2 are present. It becomes a bit harder for proteins that share some peptides. Like in this case here, there are 4 peptides, and the middle ones are shared between proteins A and B. What can you do there? Well, you can still differentiate them, but only if you have the right evidence. If you see only 2 and 3, then there's no way that you can pull them apart. There are other cases, like subset proteins. Here, there's no way you can ever prove the presence of B. Why is that? Because if A is present, then you don't see any additional peptides from B. Do you have any idea how you can still keep them apart? Say again?
If you see A, you know it's not B.
Yeah. Yeah, if you see A, you know that A is present, but what do you know about B at this point? Yeah. You can not do that with qualitative proteomics. You need to go to quantitative proteomics, and you can start comparing ratios. We'll talk about that in a minute. It's not that trivial. And then there are other things that are also difficult, like these subsumable proteins. If you see all 4 peptides, you can't really say whether you have A, B, and C present, but A and C would already explain all the observed data. And that would be a simpler explanation than saying I have A, B, and C in there, even though that might be correct. Just based on the data, you can't tell. You simply can't tell. Okay, so what I've introduced now is basically the notion of so-called protein ambiguity groups. These are groups of proteins that share peptides and that lead to ambiguity in the assignment of proteins. And if you want to be correct, what you report is not a list of proteins, but you report these protein ambiguity groups when you have them, to be sure that all the evidence that you have is included. Oftentimes your search engines will report only one protein, the one that they had a hit against, but it doesn't tell you that there have been 10 other proteins that contain peptide. So you've got to be a bit careful. So in this case here, we would have a protein ambiguity group that contains three proteins, and they basically share the middle across all of them, and then the first partnership between AB and AC. Now with parsimony-based inference or Occam's razor, what would we say? Well, we would just argue if we see these four peptides, the most likely explanation is the simplest one, namely that A is in the sample and none of the other peptides. But an educated guess. That can also be that you have B and C sample in no way whatsoever. So the reality looks a bit more complex. This here is an example where we observe 13 peptides. These are the guys here. And we have a bunch of proteins on the right-hand side. And whenever you have an arrow between a box and a circle, that means that this peptide is contained in this protein. Yeah, so peptide 1 is contained only in protein A. So if you observe that, we know something that A should be there. But the same is for peptide 2, but peptide 4 comes either from A or from C. And this way you can try to read this graph. Here 9 and 10 are contained in both F and G. And here it becomes a bit complicated. 11, 12, 13 are contained in H, I, and J. So if you do this protein ambiguity list output, what it would give you is that protein A is supported by peptides 1 and 2. Now we have seen these two guys, so A seems to be there. Protein B is fine as well. We have support for peptides 3 and 4. And now we have the first ambiguity. Here we have already a mark there that says, well, 4 also supports protein C. But we are sure that protein B and protein C are both in there because they have support from unique peptides, namely from 3 and from 5. So that is fairly simple, but then we run into cases like this, where we have a protein group. And here it says there are 3 proteins that are of interest to this question, H, I, and J, and they have support from different peptides. But at this point, we cannot say what is truly in the sample. Just that these are possible candidates explaining what we have. And that's a more honest answer than sort of randomly picking one of the 3 and hoping for the best. Getting back to this question I had earlier, you can also use quantification for inference, suggestion you had, because you know that each of these peptides contained in the protein sequence just once. So if you could accurately quantify each peptide, then that would be easy, right? What's the problem there? We can't quantify properly because we're going to do that tomorrow morning only. No, the problem is really that we can't easily quantify absolutely with peptides. But what we can do is we can look at ratios between samples, and that is often indicative. It's rarely done. It's possible. If you run into this problem, you might look at that. We can look at different ratios across samples. I will not go into that in too much detail, but fundamentally, when you have two samples that contain the same proteins, then if you compare across samples, the peptides that are related should also change their ratio in the same way. And if you see that the ratios are changing across samples in an odd way, then that usually means that you have different isoforms underneath. And that is one way to figure that out. Let me skip forward a bit because that is getting, we're running out of time. When I said earlier 1% FDR on the peptide level means that, or on the PSM level, that does not mean that we have 1% FDR on the protein level. And this is particularly true if you have large-scale experiments. Can you imagine why that might be the case? So oftentimes you see in the literature people aggregating, in order to increase the protein count, aggregating measurements from different fractions, aggregating across thousands of samples in order to find all the proteins. You remember the human proteome paper we were talking about earlier that was aggregated over a large data set. So what happens if you do that?
So then you maximum all these correct dates. The maximum is going to be very close to the distribution of the correct date.
Yes. So let's look at these random hits. Random hits occur by chance. So if you measure the same spectrum, the same peptide twice, let's assume the peptide is really not is really in there, but you have a random hit that assigns it to an incorrect protein. If you measure the spectrum again, the spectrum will be different, and you will actually match a different protein randomly. The odds of hitting the same protein over and over again is only high for true identifications, not for the incorrect identifications. They will be all over the place. So if you aggregate over many samples, what you do is you accumulate false positive identifications. So you apparently get more and more proteins, but in reality, all you get is more and more incorrect identification because the set of true peptides that you really have in your sample is going to remain the same. And that is it took people a while to realize that. They thought, yeah, I just measure more and more. I do triplicates, quadruplicates, even more than that. And they always got, of course, higher number of identified proteins, but not after FDR correction. Then this number actually goes down because you have to account with all these incorrect hits. So the trouble here really is that the number of proteins, if you map the FDR on the peptide level back to the proteins, then you can have lots of random matching, and the error rate can be a lot higher as you aggregate over proteins. And people didn't understand that initially and just thought that I have PSMs. So it is really essential that you do this FDR correction also on the protein level. And there are a few ways to do that. You might have seen that here and there. In the same way that you can control against for FDR on the PSM level, you can also do that on the protein level. And that leads to different thresholds. People tend not to like it because they lose so many proteins. Yeah? But you should keep in mind that you're losing the incorrect identifications primarily. And you get a much more accurate estimate of what you really had in your sample. OK. I'll stop here, and we continue with the hands-on, and you can try out this FDR, can try out what changing the search does to your results.